All right, friends, welcome back to the very riveting part 2 of the Dr. Hayes video lecture. I thought about changing clothes, but that seemed to be a little much for the day. As I mentioned before, my goal today is to make a video lecture of chapter 17. I will probably break this up into 2 sections just because I'm going to need a little bit of a break in between. This is not something I normally cover in one class, so getting up here and trying to do it all at once is a lot. So this will probably be broken up into 2 shorter videos. We will have some questions that we will do in class 4 points on Tuesday. So I would make sure I sent the outline for this chapter with the email that these links were in. So if you haven't, I would pause the video, go print off that outline. That way you can take some notes as we go. So in A&P2, we literally just pick up where you left off in A&P1. In A&P1, you should get through chapter 16, the endocrine system, and I'm working under the assumption that that is how far you got. So we're picking up with chapter 17, which is blood. Now, blood's really unique because it's the only tissue in your body that is a fluid. Every other tissue in your body is a solid. Now, it's connective tissue, and remember, the hallmark of connective tissue is that you have living cells, which we'll talk about the different types of those, in what's called a non-living matrix. So in the case of blood, that non-living matrix is going to be the plasma portion of the blood. Now, if we really want to boil this down very, very simply, we can say that the function of blood is to distribute things, to regulate things, and to protect things. But we're going to dive into that and kind of unpack what we're distributing, what we're regulating, and what we're protecting. So the first thing I want to ask, though, is what color is human blood? So think about that for a second. Now the answer is human blood is always red. Always, no matter if it has oxygen in it, if it doesn't have oxygen in it. And if you're like me, you probably learned in school something along the lines of blood is blue until it hits the air and then it turns red. And that is actually one of the most common misconceptions about the human body that there is. Blood is always red. And you can see down here there's a link to a little video. You can go in and watch that video. It's a great video made by the Crash Course people. If you're not familiar with Crash Course, it's a lifesaver. Big pet peeve of mine. Blood is always red. And just to give you an idea, usually when I poll the class on this question at the beginning of the semester, usually about 75% of the class says blood is blue when it is deoxygenated. Now that brings up an interesting point of why would our vessels look blue if the blood in them is actually red? And that's just simply has to do with how well wavelengths of light can travel. Red wavelengths of light can't travel very well, and so we don't get a red appearance of our blood vessels. And I think we would look kind of weird if they were red. So on the distribution portion of blood functions, we're going to distribute a couple of different things. One of those is oxygen. We're going to spend a great deal of time talking about hemoglobin and how oxygen moves through the blood. So we're going to distribute oxygen as well as nutrients, and those are the nutrients that your digestive system absorbs from your food, and those 2 things are going to go to the cells. Now remember, the reason those things go to the cells is for our body to do cellular respiration. Our body needs oxygen and glucose to do cellular respiration, which produces ATP and carbon dioxide. Now, carbon dioxide is one of these metabolic wastes that gets produced by cell processes. It's not the only one. Other things like ammonia, nitrogenous waste, those things also get transported to the blood and taken to whatever organ is going to help eliminate them from the body. So in the case of CO2, that waste is going to go to the lungs. In the case of nitrogenous waste, it's going to get filtered out by the kidneys. Also, blood is responsible for distributing or transporting hormones. If you'll remember from chapter 16, hormones are made in one place, an endocrine gland or cell, and then transported via the blood to wherever their target is. Now, in terms of regulation, one of the big things that blood helps regulate is your body temperature. And I know that seems very strange, but if you remember all the way back to the chapter in which you discussed the integumentary system, we said that underneath our skin we have this network of blood vessels called the cutaneous plexus. And at any given moment, a lot of your blood is associated with those blood vessels. Now, the way they help regulate body temperature is blood vessels can dilate, get bigger in diameter, or constrict. And so when you're hot, think about what happens. Sure, you sweat, but you also tend to get really red in the face and flush. If you put your hand up, you can kind of feel heat radiating off. That's because when you're hot, those blood vessels dilate, moving that hot blood toward the surface of your skin and allowing that heat to radiate off. When you're cold, on the other hand, those blood vessels constrict, and it keeps the warm blood more toward your main organs, right? Think about what happens when you get cold. Your toes and your fingers and your nose and your ears go numb. That's because we're moving that blood flow more toward your vital organs. Now, while you might look different with no nose or toes or ears or fingers, you're going to be alive. But if your core temperature, if your organs get too cold, you wouldn't be alive. pH and fluid volume are going to be huge roles that the blood have. In fact, we will spend an entire chapter later on really focusing on how the body maintains the right amount of fluid, as well as how the body maintains its own pH. Now, on the protection front, this is going to focus on preventing blood loss. If there's damage to a blood vessel, we have got cells and cell fragments in the blood that will keep you from bleeding to death, as well as cells that are going to be involved in preventing infection. We'll spend an entire chapter talking about the immune system and how those cells function. So it's really interesting because we tend to come back to these concepts in the blood chapter over and over and over throughout the course of the semester. Now, if we look at blood, and one thing I'd like to draw your attention to, this right here, anytime you see this, that's telling you what page that figure came from out of the Mary-Ebb book. But that is not the most recent version. This is the version that has kind of a maroon or brick red spine on it. The new edition just came out and I have not updated those yet. My guess is it's within a couple of pages and the picture is probably very similar to the new edition of the book. So if we look at what's going on in blood, we said that blood's a connective tissue. So that means we have living cells and we have a non-living matrix. And blood is what's classified as a suspension. And that means if we let blood sit or we put blood in a centrifuge, the cells are going to settle out because they're heavy and dense. This is different than a solution, say sweet tea. If you make your sweet tea and you let it sit, all the sugar's not going to fall out to the bottom because the sugar particles are really small. Here, because the blood cells are really big, they settle out toward the bottom. So we're going to spend a little bit of time talking about the plasma as well as the different types of what we refer to as formed elements. So that's going to be our leukocytes, or white blood cells, our platelets, and our erythrocytes. So plasma is mostly water, 90% water. And you can see a little bit better here in some real blood. We have this very large plasma component, it's kind of this yellowish color. While it is mostly water, there's lots and lots of stuff dissolved in that plasma. So these are really small things like electrolytes, or some bigger things like our plasma proteins. Now, electrolytes are by far the most abundant. There's lots and lots of them. And remember, an electrolyte is an ion that has a charge. So we're talking sodium, chloride, magnesium, potassium. There's several others, but those are kind of the big kickers there. We also have proteins in our plasma, and while there's not as many proteins as there are electrolytes, proteins are really big, so they're the most by weight. So these are the different plasma proteins that we find in the blood, in humans, of course. These are all made in the liver. So albumin is the most abundant one of all of them, and there's this great table on page 633 that kind of goes over the components of plasma. Albumin is going to do a couple of different things. I think I have a separate slide on it. I think I'm getting ahead of myself. We also have globulins. There's a couple of different kinds of globulins. And then fibrinogen, while fibrinogen is not very common, it's going to play a really important role in blood clotting. So if you eat eggs, you have eaten albumin. Albumin is the white of the egg. We also have albumin in our blood. And one of its big roles is to act as a shuttle or carrier protein in the blood. So this picture over here I've actually taken from an advertisement for a pharmaceutical, a drug, paclitaxel. I'm not exactly sure what it's for. The problem is, because blood is mostly water, things that don't like water have a hard time moving through the blood. If you'll remember back to previous science classes, we have stuff that is hydrophobic and hydrophilic, right? Hydrophilic means water-loving, hydrophobic means water-hating. And the problem is if you have something hydrophobic, something that doesn't like water, it often has a really hard time moving through the blood because the blood's mostly water. So what happens is these hydrophobic things like pharmaceuticals, like hormones, will attach to albumin, and albumin will shuttle them through the blood to wherever they need to go. Now, albumin is also going to play a role in buffering, so that's going to mean maintaining the pH of the blood, and it's also going to play a role in maintaining osmotic pressure. We're going to spend a lot of time talking about those other two in much later chapters. Now, globulins, there's three kinds: alpha globulin, beta globulin, and gamma globulin. Alpha and beta act really similar to albumin. Their job is to transport substances through the blood. Gamma globulin, however, is actually going to act as part of the immune system, right? It's going to act like an antibody, and we'll spend a lot more time talking about it a little bit later. And then fibrinogen, again, these are all produced by the liver. I know I make it sound a little different there. Fibrinogen is critical to blood clotting. Blood clotting is an incredibly complicated process with lots of individual steps, and if any one of those steps gets messed up, you may not have appropriate blood clotting. Now, we also mentioned that there were electrolytes. The reason we have so many of those is they're going to help with that osmotic pressure and pH. Again, we'll talk a lot more about that later. And they include all these different things here: sodium, potassium, calcium, magnesium. Bicarbonate is a really big one that we'll spend quite a bit of time talking about. That's HCO3 minus. Unfortunately, I can't write and talk at the same time on the screen. Now, we know that a lot of sports drinks advertise themselves as being high in electrolytes, and that's true. The real downside to sports drinks like this is they're also usually very high in sugar. So you may not really get the benefit that you think you're getting. And for most of us that are not super hardcore endurance athletes, usually we can get by just fine with water. But often in the summertime when it is hot outside and you're sweating a lot, an electrolyte replacement can be really beneficial. But there are some other things. There are these little electrolyte gels that bikers and runners use. Coconut water also has a lot of electrolytes in it. You just have to be really careful because a bottle like this is probably 3 to 4 servings of Gatorade and has a pretty significant amount of sugar in it. So let's talk about the formed element part. right? This is going to be leukocytes, platelets, and erythrocytes. And the really interesting thing about these is they don't last very long. Most of them only last from just a handful of days up to maybe a month or two, depending on the cell type. And they don't actually divide. You spend all this time in intro bio and in A&P1 and you talk about cell division and mitosis, and then I blow your minds and tell you that none of these cells actually do cell division. What they do is we have got what are called stem cells in our bone marrow. And so, say an erythrocyte needs to be replaced, rather than an erythrocyte dividing and making a copy of itself, the stem cells in the bone marrow will form a new erythrocyte. And we're going to talk about each one of these. There's definitely some similarities, but also some pretty big differences between them. Erythrocytes are by far the most abundant. They're the heaviest, they're the densest, they fall out to the bottom of that blood sample. What's super weird about them is this. They don't have a nucleus, they don't have organelles, and that means they don't have any DNA. So if you are getting a DNA test done, say a paternity test, and they were drawing blood for it, or they found blood at a crime scene and they were trying to match DNA with someone, they're actually not getting the DNA from the red blood cells. It's a big difference. Big difference is our white blood cells do have a nucleus and they do have DNA, which is why if you're doing some sort of DNA test, whether it's the Ancestry DNA or 23andMe or a paternity test, they'll often use a cheek swab and get those cells because they do have DNA in them. Now I know that's really weird, right? What kind of cell doesn't have DNA? This is pretty unique to humans. Lots of, mammals. Reptiles, amphibians, they have nucleated red blood cells. We don't. I'll show you how that happens. What an erythrocyte does have in it is a lot of hemoglobin, some antioxidant enzymes, and some proteins. And really the main goal, the entire goal of an erythrocyte is to transport oxygen throughout the body. So it's a pretty unique structure. I always use the analogy that an erythrocyte to me looks like you've taken a Junior Mint or a Mentos and kind of squished it in the middle. There's this indentation, this kind of concave shape to the erythrocyte. And the reason for that shape is it gives really high surface to volume ratio. And what this means is it's very easy for oxygen to diffuse into and out of that cell. And it also leaves a ton of room for hemoglobin. Because there's no mitochondria or ribosomes or nucleus in there, we can fill this erythrocyte with hemoglobin. And what's even crazier is it doesn't do regular cellular respiration. Because these erythrocytes don't have mitochondria, which is where cellular respiration happens, they actually have to make their ATP anaerobically. So even though there is a lot of oxygen present in erythrocyte, it doesn't use any of that oxygen to make its own ATP because it doesn't have a mitochondria. So let's talk about hemoglobin. This is what hemoglobin looks like. It's a protein, and it actually has kind of 4 sections to it. So there's a section here, 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 and here. This is its tertiary structure. So it's got 4 little sections to it. It also has associated with it these 4 red things here. These are what are known as heme groups or heme pigments. That's H-E-M-E, heme. And then in the middle of that heme is an iron, right? So we've got our globin protein, which on this figure is the brown stuff. We've got our heme group, which is the red, and then we've got our iron in the middle. So each hemoglobin molecule is capable of carrying 4 oxygen molecules. So that's O2. We can have an oxygen attached to each one of those heme groups. Now, not every species uses hemoglobin. There are other different, and hemoglobin is why our blood appears red. The iron in that heme group gives our blood that red color. Occasionally it's really bright, occasionally it's a little darker red, but that's where the red comes from. Other organisms, though, can use hemocyanin, which gives them blue blood, hemoerythrin, which gives bright pink blood, biliverdin gives green blood. So mammals use hemoglobin, but other groups of organisms, vertebrates and invertebrates, can have different colored blood depending on what is being used to transport oxygen in their body. Now, here's what's crazy, is there are millions and millions and millions and millions and millions, if not billions, of hemoglobin molecules in your body. So an individual erythrocyte, while each hemoglobin can only carry 4 oxygen molecules, there are so many hemoglobins, we're capable of carrying a ridiculous amount of oxygen at any given time. So there's a great video that is on Georgia View about hemoglobin function, but I'm actually going to pull it up real quick and see how it looks on this video. So let's take a look at that real quick. If I can work a computer. Well, this appears like it doesn't want to give me any audio, so let's just take a look here and I can kind of narrate along to this video. So we see our hemoglobin here, and you can slightly see there's one of our irons, there's one. I don't think you can see the other one just due to the angle. So let's take a look here. Remember, that's the globin protein. There's an oxygen molecule that is coming, and it is binding with that iron-containing heme group. Now, what I hate about videos like this is it kind of showed that hemoglobin looked like it changed color a little bit. Blood does not change from red to blue, but it does change a little bit from a dark red when it's deoxygenated to a very bright red. If you think about when you get a paper cut, how bright that blood is versus when you have blood drawn out of your arm, it's often really dark. So what they were showing you there was just the erythrocytes moving through the bloodstream, single file, in order to do that. And hopefully by the second part of this video, I will have some audio to show you. Those videos are all posted on Georgia View for you. Now, when hemoglobin has oxygen attached to it, we refer to it as oxyhemoglobin. Now, that could mean it has 1 oxygen, it could mean it has all 4, but we would call it oxyhemoglobin. If it does not have oxygen attached to it, we would refer to it as deoxyhemoglobin. Now, this is what's really interesting and another very common misconception about the body. Often people think, okay, I pick up the oxygen, I take it and drop it off, and then I pick up a carbon dioxide and carry it back to the lungs. And hemoglobin can carry carbon dioxide, but it doesn't carry it in the same place. So if we go back, we said that the oxygens attach here to the heme group. If there were CO2 attached to the hemoglobin, it would attach out here to the globin protein portion. But one thing you are never going to see is both oxygen and CO2 attached to the hemoglobin at the same time. It will only carry one or the other. Now, the question is, how do these erythrocytes get formed? They don't do mitosis the way that other cells do. So when we're talking about the formation of blood cells, we're going to call it hematopoiesis. And that's a general term for any blood cell formation. When we're talking about the formation of red blood cells, we're going to refer to it as erythropoiesis. And I kind of hate that word poiesis because the I's and the E's just never seem to be in the right place. So this process is going to occur in the red bone marrow. And we know in adults, red bone marrow tends to be concentrated in the sternum, as well as in the ball of the femur and the joints of some of the larger bones. And we're producing about 100 billion new cells every day. That's pretty remarkable to think about how many cells there are. Now you'll notice over here, here's our stem cell. This is referred to as a hematopoietic stem cell. All of the cells in your blood, erythrocytes, leukocytes, and platelets, all originate from the same exact stem cell. It is capable of becoming any one of those three types of cells. But you'll notice the next step in this process is referred to as a committed cell. Once that stem cell transitions to a committed cell, there's no turning back. Okay? Once you're to that point, you cannot change your mind and go, no, I was just kidding, I really wanted to become a platelet. Not an option there. So all formed elements, erythrocytes, leukocytes, and platelets all start as a hematopoietic stem cell, and then they're going to become a committed cell, and there is no turning back once you're to that point. And you'll notice the erythrocyte has a nucleus at this point. It has organelles. So during this really early developmental stage, our cells are going to produce a lot of ribosomes. And we know that ribosomes are responsible for making proteins. So this is where our hemoglobin is going to come from. If we want to have a ton of hemoglobin in this erythrocyte, we're going to have to make it back here. So this is where the hemoglobin is getting made. We're doing a lot of division. We're accumulating that iron. Now it's important to note the body cannot make iron. That iron's having to come from your diet. So if you've ever heard somebody referred to as anemic, or you are anemic, that often means you don't have enough iron, and that can affect the number of red blood cells that you have. Now, you'll notice once we get further down in this developmental pathway, the organelles and the nucleus are gone. So we kick the organelles out, the nucleus degrades, and it starts to get closer to the shape that we're familiar with an erythrocyte. So at this point, once those organelles are gone, you can't make any more proteins, you can't divide anymore, you are what you are. Now, a very young erythrocyte is referred to as a reticulocyte. This is the very last step before we have a fully functioning erythrocyte. This is just the last little step of maturing before you're ready to start carrying oxygen and doing your job. It's kind of like when you get a new job, you have to be a trainee for a little while. I feel like that's kind of the stage that a reticulocyte is at. Now, the question is, what controls this process? How does our body know when it needs to make more erythrocytes? We said on average, it makes something like 100 million a day, but sometimes you need more and sometimes you need less. So a hormone is what is going to control this process. The hormone is known as erythropoietin. I usually abbreviate it, and books usually abbreviate it as EPO. This hormone is going to stimulate the red bone marrow to make more erythrocytes. And this hormone is mostly produced by the kidney. The liver produces a tiny bit, but really the kidney is who's in charge here. Now the question is, something has to stimulate this. As with most hormones, this is going to be a negative feedback process. So if you remember this seesaw thing from AMP1, it's all about homeostasis. right? There is an appropriate level of oxygen in your blood. And if that level gets too low, your body is now out of homeostasis. So the body will respond to try and fix it. So the stimulus here is what's referred to as hypoxia. And this basically just means you don't have enough oxygen getting to your tissues, right? Low blood oxygen. And it can be caused by several different things. One is just if you don't have enough red blood cells, you can become hypoxic. If you don't have enough hemoglobin, you could become hypoxic. Or if not enough oxygen is available, you could become hypoxic. Now, it's interesting to think this sounds like a very dramatic situation. But for someone who has anemia, they may be experiencing one of the first two. For someone with kidney issues, they could be experiencing decreased red blood cell count. If you're really sick, say you have bronchitis or pneumonia and you're really congested, maybe you're not bringing in enough oxygen. Say you fall in a pool and you start to drown or you get strangled or something really dramatic, you wouldn't have enough oxygen. If you go somewhere with high altitude, you go to Denver or Mount Everest or wherever you go, there's less oxygen available in the air and you would probably become hypoxic. So this isn't as quite a dramatic situation as it sounds. You can become hypoxic just in the course of your everyday life. So it doesn't have to be some dramatic illness to get you there. So all of a sudden we're out of homeostasis. The amount of oxygen in our blood is too low. We are hypoxic, and that is going to trigger the kidney and a little bit by the liver to release this hormone erythropoietin, EPO. That EPO is going to move through the blood. Remember, hormones made one place, act somewhere else. And its target is going to be the red bone marrow. So hypoxia caused by whatever situation has triggered EPO release. That EPO is going to act on the red bone marrow, and it's going to tell it, I need you to make more erythrocytes. Right? So we see enhanced erythropoiesis. The red blood cell count is going to go up. And when the red blood cell count goes up, that means our blood is capable of carrying more oxygen. So all of a sudden, our unbalanced seesaw, our blood oxygen, which had gone way down, is going to start to come up and start to come up and start to come up. And eventually, when that oxygen level in the blood is back to normal, this process will stop because you would no longer be hypoxic. So remember, the key with negative feedback is you're out of homeostasis, your body responds to fix it, and once you're back, that response would stop. So if we were to take a blood sample from someone and see high amounts of EPO in their blood, that tells us that this process is occurring and that they're in the process of making more erythrocytes. Now, interestingly, pretty much, I wouldn't say every single thing the body makes, but most of the things the body makes can be found in exogenous forms. So when our body produces EPO, we refer to that as endogenous. But exogenous EPO would be like something you get from a pharmaceutical company and you are putting into your body. So for example, we make endogenous insulin. A type 1 diabetic uses exogenous insulin. So EPO is on the market for patients who have too little EPO. We often see this in patients experiencing kidney failure that are on kidney dialysis. They're not producing enough EPO, and so the number of red blood cells or their hematocrit is too low. And so they'll take EPO injections to try and keep that red blood cell count up. Now, interestingly, as with most things, people can find a different way to use it. Some athletes will use EPO supposedly to enhance performance. And the argument is that if I increase the number of red blood cells I have, I can increase the amount of oxygen I can carry, and I can perform at a higher level. The problem with this is if you increase the number of erythrocytes outside of the bounds of normal, you can start to increase the risk for blood clots. And so there's a definite risk of using exogenous EPO when you don't have a medical condition. So I mentioned before that our body is not capable of making iron, that iron has to come from your diet. And most of the iron in our body is located in our hemoglobin. The rest of it is actually going to be stored in the liver, which is why, just as a side note, my dad's favorite meal was fried liver and onions. A lot of people eat the liver of different organisms, but the reason that organ meats in general were often eaten so much is because they're super nutrient dense. There's a lot of iron in liver. I do not enjoy liver. That is the one meal when I was a kid that we were allowed to have peanut butter and jelly sandwiches. Normally we would get this is dinner, and if you don't like it, breakfast is in the morning. But that one meal, I was allowed to eat a PB&J because my mom didn't like it either. Now the tricky thing about iron is it is actually toxic to us if it's in its free form, if it's not attached to something. And so our body will attach iron to these different proteins. We have ferritin and hemosiderin. That way the iron can be stored without harming us. Now, if we need to move the iron throughout the body, we use a different protein called transferrin. That one's pretty easy to remember because it sounds like transfer, right? Transferrin would help move that iron. Say we were making some more blood cells and we needed some iron, it would have to move from our liver to our bone marrow. Now, an erythrocyte can last a couple of months, 100 to 120 days. And part of the reason that this is so short is you have to remember an erythrocyte has no nucleus and no organelles, which means if a cell becomes damaged, it doesn't have the means to repair itself at all. And often what happens when an erythrocyte starts to get older, starts to get toward the end of its lifespan, what we usually start to see is loss of flexibility. Erythrocytes are very, very flexible, and that's incredibly important to their function because when they get down to the tiniest of the tiny capillaries in your body, those capillaries are so narrow, the erythrocytes have to go single file, and they kind of have to do this. They're like squeezing and bending their way through those capillaries. So if they lose flexibility, it can mean they can't get through those capillaries efficiently. And hemoglobin will also start to degrade and become less capable of carrying oxygen. So while you may have enough hemoglobin, it's starting to become less and less functional. So this was about the formation. This is a great figure. It's in your book. It's a long figure I've kind of subdivided. This part right here covered how the erythrocytes were made, which we already talked about. But then the question is, what happens to them once they've hit that 100, 120 day mark? They're not flexible anymore. They can't transport oxygen. The cool thing that our body does is it often tries to recycle components before it just outright junks them. And I use lots of analogies. The analogy I like the most here is think about what we do with cars, right? Even a car that's been in a wreck will often wind up at a junkyard or a pull apart. So even though that car might have been wrecked, say somebody needs like an interior console for a 2005 Buick. Well, the car might have been wrecked, but there's nothing wrong with the console or the rear view mirror, or they need a seatbelt assembly for the rear passenger side seat. So what we do is we'll often go pull off the parts that are still useful, and then we'll get rid of the rest. And that's what our body does often when it needs to dispose of something, and erythrocytes are very similar. So this is going to happen a couple of different places. The destruction of an erythrocyte is going to occur in the spleen, which is an organ over here on your left side, the liver, and the bone marrow. So in all three of those places, we have cells called macrophages. We're going to talk about them more in the immune system chapter. But macrophages are these large cells capable of engulfing and destroying something. So a worn out erythrocyte is going to get engulfed by a macrophage in either the spleen, the liver, or the bone marrow. And the hemoglobin is really the part that's going to get broken down and salvaged. And remember, an erythrocyte is 97% hemoglobin. So notice here, our hemoglobin is going to get split into two parts. One of them is the globin, that's the protein part, and the other part is the heme. So the globin protein is just going to get broken down into the individual amino acids, and those are going to get put back into the blood. If you remember, proteins are a lot like Legos, right? You've got 20 different amino acids that are assembled in different orders. Those proteins fold up and have different functions. Well, if you think about playing with your Legos when you were a kid, you didn't build a princess castle and then just throw it away. You built a princess castle today, took it apart, and built a pirate ship tomorrow. So we can just disassemble this protein and use those amino acids to build a different protein later. The heme group is divvied up into two parts. One is the iron, and that's going to get stored in the liver, right? Remember, it's stored attached to either ferritin or hemosiderin. And then the other part of the heme group is known as bilirubin. It's a fun word. I like it a lot. We actually had someone get a package delivered that said bilirubin, and somebody thought it was somebody's name, Billy Rubin. Kind of entertaining. So that bilirubin is going to get picked up by the liver, right? And then it's going to get secreted into your intestines. Your liver is what's responsible for making bile, which is part of the digestive process, and it'll just mix that bilirubin into the bile and dump it into your intestines. And what's really interesting is the bacteria that are in your large intestine will actually break down that bilirubin into a substance called stercobilin. That is what makes your poop brown. So the breakdown of erythrocytes is what is responsible for the color of your feces. Who knew? Excuse me. Now, you may have heard of bilirubin before. That's what's responsible for jaundice if it is not disposed of properly. So my niece, a lot of babies, even if they're full to term babies, sometimes babies can have a little bit of jaundice when they're born. It's because there's a little bit of a lag in this breakdown process due to the bacteria. So they'll use what's called a Billy light sometimes. They'll put the babies under this little light that's the right wavelength to help break down the bilirubin. It's the same substance that's causing that jaundice. Now, I am actually going to pause right here so I can take a little bit of break for my voice, and you'll just come back and there will be a separate link for part 2 of this chapter. We don't have that much more left to do.